today's podcast is on how exercise affects the brain in good ways. And my guest is Christiane Wrann, and she's I don't know if you're assistant or associate
Professor.
Associate professor at Harvard. And she is actually from Europe originally. Germany. And there you got your veterinary medicine degree in Germany, and then you went on from there. But why don't you talk about that? Because you were initially interested, you'd planned on becoming a veterinarian, and you still are on paper, but you kind of took, rather than seeing and treating animals, you're doing research to try to, well, to help humans now, but also, I guess, help animals. We could talk about that a little bit. So welcome.
Yeah, thank you very much for the invitation. It's a pleasure to be on the podcast, and I'm looking forward to our stimulating conversation. So yeah, I'm originally from Germany. I studied veterinary medicine. So I was a competitive equestrian in my youth, and I kind of got intrigued by, horses getting injured in competitions, and lack of treatment, and trying to understand why some horses got injured and not the other ones, and how do you diagnose and treat? So that was kind of the idea to, what the motivation, what drove me to study veterinary medicine. And then during my studies, I really understood that to kind of, increase our ability to, understand and cure diseases, you have to go beyond just the clinical work. You really have to go into the research part. And that's why then I decided to do a PhD and, got really into what I think now we would call molecular medicine, to understand which are the molecules that are, acting in exercise and how they're acting, and, how that's, in this case, we're going to talk about the brain, what are the effects. So, and then after my, finishing my PhD, I went to Harvard and did a postdoctoral training at Harvard Medical School, and then became an assistant professor at Mass General Hospital, Harvard Medical School. And, now, as you said, I'm associate professor and the leader of the program in neuroprotection over there.
But before I started this podcast, I was talking to Christiane about kind of some overlap in our backgrounds, because I used to train and race harness horses, trotters and pacers, and she, as she mentioned, did equestrian ride. What did you do? Jumping or
dressage riding, and what we call a quadrille. So this is the formation where you have like a team of 8 or 12 people, and you do to music. You're doing formation.
Oh, it's like synchronized.
Synchronized swimming, or or like dance. If you just think about the dance performances that they have, like where you don't have the solos of 2 dancers, but you have a whole team that's performing. So we did that with horses, and it was great fun, because you had also the team aspect there. You had the competitive eventing aspect in there, and I'm still a huge fan of horses. They're beautiful animals. They're fascinating.
Amazing.
Yeah.
And one thing as far as the effects of exercise on the brain that I think anyone who's had horses will probably appreciate is that if they can't get exercise, they get ornery and cranky. And
Yes.
if they do get exercise, it improves their mood. And that's also true in humans. It's true with me. Exercise was really my kind of release for anxiety, and I've had some injuries actually recently and can't exercise, and like my mood goes way down. And so we'll talk about what we think is happening in the brain with that, and also cognition. But you started out in immunology, right? Some of your first work was looking at the immune system.
Yes, so I did my PhD in immunology because at that time, immunology was the field that was most advanced to looking at molecules. They already had identified a lot of these, these CD markers, so you could distinguish these different subtypes of immune cells, using FACS analysis. So They had very nicely mapped all the differentiation cascades of how the immune cells. So I really felt that was a field where you had already, good anchors on the molecular level. And then, as it turns out now, actually a dysregulated immune response is causative in many diseases, not just in infectious diseases or autoimmune diseases, as maybe some people thought originally, but neuroinflammation and neurodegenerative diseases. Aging in itself, in a lot of times can raise chronic low-grade inflammation in different tissues. If you think about, obesity and, type 2 diabetes accompanied by a chronic low-grade systemic inflammation. So, we can almost ask the opposite question, like, are there any diseases now where we think the immune system is not involved in some at least in some shape? And that, it makes sense. The immune cells are everywhere. And also, I think our understanding of what does actually, inflammation mean? It's not just in, maybe originally people were thinking about, oh, it's the infiltrating immune cells, neutrophils, monocytes, or T cells. But we now also understand that a lot of the tissue cell types like hepatocytes, adipocytes, or neurons in itself can secrete certain, mediators to talk to immune cells. So it's not just immune cells among themselves, it's the crosstalk of.
Yeah, one kind of the main reason that so much was learned about at the molecular level about the immune cells is a practical thing. You can just take blood particularly from human research, right? You can just take blood and you centrifuge, and there you got these cells. Brain can't do that with humans.
Yes, and then they're also they're a nice single cellular suspension, right? You don't have to dissociate anything. They're already in the state that you want. So yeah, it definitely made it more accessible. And I think that's something on the flip side, what also then later on drew me into the neuroscience was the complexity, right? Looking at the brain, it's such a different animal, to kind of, pun intended here, than looking into that. And that's something that I really came to appreciate in my training, because I did start out working also a lot with peripheral tissues, muscle, fat, a little bit liver. And they're just, I don't want to say one-dimensional because I don't want to do anyone injustice working in that area. But if you think about the brain, where it's not just you have all these different cell types, the neurons, the different glial cell types, blood vessels, but also the cellular organization in the hippocampus, in the dentate gyrus, the CA1, the CA3 is very different than you think about the cortex, but they're still talking to each other. So there's a very different layer of complexity in the CNS, in the brain, which, I guess for people who love a challenge, it's a draw. Sometimes for the students or us in the lab making it work, it can be a pain, but it's also the beauty.
One thing that was interesting historically is that in immune cells, lymphocytes, particularly all these cytokines and signaling pathways were discovered, the interleukins and tumor necrosis factor and so on. And then later on, it was found that neurons, can respond to these, or even in some cases produce these. And then if you look from an evolutionary perspective, actually organisms had nervous systems before they had humoral immune systems.
Mm-hmm.
It was probably innate, the macrophage-like cells might have preceded. So probably some of these signaling pathways were in nervous systems evolutionarily before they were in the immune. Because we did work with TNF, and we had this finding that we had a lot of trouble getting the paper published because we did a bunch of studies with neurotrophic factors, BDNF, NGF, FGF2, and found they were neuroprotective in certain models we had, cell culture. And then we started looking at pro-inflammatory cytokines, and we found that TNF, if we pre-treat neurons with TNF and then hit them with excitotoxic insult or metabolic insult, they're actually protected, and it's through this NF-kappa B pathway. But anyway, these these cytokine things, they do a lot of good things. I think even in the absence of infection or, any disease state, they're serving normal functions in the nervous system.
Yes, that's true. I mean, there's a surveillance. There's also like a lot bigger appreciation now of the importance of the immune system, also for tissue regeneration. Getting back to the
Yeah.
exercise, we do know that part of the training effects in exercise, specifically in the muscle, but or skeletal muscle system, is by causing micro injury that then gets repaired, and then you end up with a stronger muscle or stronger tendons than before. And part of that is the immune response. And I think it started out with this. There's this. Think about the M1, M2. Like there's a anti-inflammatory, like a pro-inflammatory, anti-inflammatory immune response. I think now we think more about like a regulatory immune response. But again, it shows you how important it is to have both. And I did my PhD work in sepsis, which is basically where you have an overregulation, and that it was kind of very clear that if you don't have a good immune response, you get overrun by infection, and we know that from congenital immune deficits. But on the other hand, if you have too much inflammation, what happens in sepsis? I think a lot of people learned about it like during the COVID pandemic, when also it was clear that a lot of the late, what ended up, people get them into the ICU was like the systemic inflammatory response. So this, the immune system overreacting, and that leading to multi-organ failure. So I think that's, again, similar to the brain. There's this balance.
Yeah.
If you don't have enough, or if you have too much, you need both. And if you think about, maybe looking forward a little bit about the treatments in Alzheimer's disease or neurodegenerative diseases, there is work going on trying to get at how do we regulate the immune system or the neuroinflammatory response, but it's always difficult because it's a dynamic system. So you don't want, if you come in too early or too late, the same intervention might be beneficial or not. There's some nice work on IL-10 in Alzheimer's disease, where you would think, oh, it's an anti-inflammatory cytokine. It would be great to have more of it, but it should, if you do it too early in the disease, actually they couldn't, like the immune cells, or especially microglia, couldn't respond to plaques. So you actually got worse disease outcomes. So
yeah.
it's finicky. And I guess that's, sometimes you get asked, oh, if inflammation is so important, why there are not better drugs already, like taking aspirin or, ibuprofen, why doesn't that protect you then from dementia? And I think the answer is it's very complex. You have to get to the right cell type at the right time. And there's, good research, if you look at it, especially, good people working on microglia, where I think we will see hopefully some breakthrough discoveries, but it's been a long way to understand how to get to the right cell at the right time point.
Okay, and then, so getting back to exercise, one of your big findings was, it was a little over 10 years ago, and you reported these really exciting findings about a molecule released from muscle cells in response to exercise that has beneficial effects on the brain. That was, I guess, when you were with Bruce Spiegelman, you were working with him?
It was during my postdoctoral training in the final years.
Yeah. So can you kind of go through that kind of as it happened?
Yes, as it happened. So when I started my postdoctoral training with Bruce, they had identified this novel exercise hormone that they called Irisin, and they had shown it was released from skeletal muscle. They also knew that it was regulating metabolism, and there was a lot of excitement about this hormone. What does it do? And I, one thing I was thinking about, and we knew about the beneficial effects of exercise on the brain. And we know that Irisin was carrying like part of the beneficial effects of exercise. So I was really interested in finding out, is Irisin at least one of these important mediators of exercise in the brain? And that was kind of the hypothesis that led me into the work. And one of the first experiments that I did is I just collected a ton of tissue, from the mice, and I looked at Irisin expression. We saw not only that Irisin was very high in skeletal muscle and the heart, but also very high in the CNS and in the brain and in the spinal cord. And then in the next step, we show that actually exercise increased irisin expression in the hippocampus of mice with exercise. We used like a running wheel system, and that really showed, oh, there was this first link of irisin also connected to beneficial effects in the CNS. And when we did a lot of more mechanistic work of how irisin is regulated, and the last very important piece in the paper is that we gave irisin peripherally, or to be more accurate, we at that time used FNDC5 because we couldn't just make the irisin part. So we used FNDC5, the precursor protein, and we expressed it with an adenovirus in the liver of these mice. And then what we could show is that this irisin got released. We found an irisin in the circulation. You don't find FNDC5. It's a type 1 membrane protein, but we found irisin in the circulation. And we also saw that we saw the same gene expression changes in the hippocampus with irisin, peripheral irisin delivery that we saw with exercise. And we had done some cell culture experiments where we either added or removed irisin, where we also saw, this neuroprotective program related in the brain. And that kind of closed the loop of saying, irisin itself or irisin talking to a factor X is responsible for at least part of the beneficial pathways, including, we're looking at BDNF. That was one of the pathways we looked in that paper.
So this FNDC5 it's a membrane protein, and
yes,
irisin is like on the outside
yes,
part of the cell, and then there's an enzyme that cleaves.
yes. We think it's an enzyme. The enzyme has not been identified, but it looks like so we have expressed FNTC5 in a whole variety of cells, and all the cells that we looked at, muscle cells, neurons, fat cells, 293 cells, CHO cells, they were all able to cleave irisin. So it must be a very well conserved enzyme. And in my postdoc we did some 4IP experiments to look which could be the enzymes. Could be, like good candidates would be, for example, the, metalloproteinases. And it would be really interesting to know if this step is also regulated by exercise. So we know that on a transcriptional level, irisin or FNDC5 expression is upregulated. And we also know we find in the circulation more irisin in mice and humans. But it might be interesting to find out if the cleavage itself is also regulated by exercise, not from a therapeutic point of view, but like at least from a, basic biology, how does it work point of view.
Is the cleavage site within the transmembrane region or outside?
Outside, just outside. And
we did some work on proteolytic processing of amyloid precursor protein, and there's a enzyme called alpha secretase that actually cleaved right in the middle of the amyloid beta peptide sequence. But anyway, it releases this secreted form of APP, and we found that had neuroprotective effects, but it was I don't think it's related to irisin because the mechanism is by activating a potassium channel and hyperpolarizing the membrane. So now I'm getting into the science. My brain's kind of running here. Has anyone recorded, put electrodes in neurons, say in culture or in a brain slice, and then record and add irisin and see if there's any effect on excitability?
So we have done kind of the opposite. We used brain slices from FNDC5 knockout mice, or again, to be more exact, we knocked out FNDC5, the FNDC5 gene at the exons, which will, correspond to later on what's the irisin protein. And if you look in the dentate gyrus of these mice, they actually show a decreased LTP formation in there. I don't remember if we also did it with irisin on the slices, because I think it was done in collaboration. I now have actually a very talented postdoc in my lab who actually is an electrophysiologist by training. So now talking to you, now my as you said, your scientist brain gets turned on, my PI brain is getting turned on. So maybe that would be a good time to revisit some of these experiments to find out what it does, especially to look at acute versus chronic effects. I think that's always an interesting question. That's coming up.
And right, so you mentioned long-term potentiation, which is a strengthening of synapses that's thought to be maybe a basis of learning and memory. And then you actually, in your cell metabolism paper in 2013, oh, there was something else in there. So we did some work on what's called mitochondrial biogenesis. Mitochondria can proliferate, and there's this, as you know, this master regulator of mitochondrial biogenesis of the regulator of the various genes whose gene products are involved. It's called PGC1 alpha. So you want to mention that before we
Yes.
move on? Yeah.
So PGC1, as you mentioned, is this master regulator. It also has been shown, among others by my mentor Bruce Spiegelman, that it regulates a whole host of the exercise programs in the skeletal muscle, including Irisin. This is how they originally found it, by looking in the mouse that is what was transgenic to overexpress PGC1 alpha in muscle, and they did it, and then they found that FNDC5/Irisin was one of the proteins most regulated by this. And then we showed in neurons on a mechanistic level that it was actually PGC1 alpha in combination with ERR alpha, because it's a transcriptional coactivator, so it needs a transcription factor, which was changing FNDC5 and expression. And we also showed in different experiments that different conditions that would reduce PGC1 alpha activity reduced FNDC5 levels, and vice versa. So
in the mice that have higher levels than normal of PGC1 alpha in muscle, do they have improved endurance?
Yes, they do have improved endurance running. So what they showed is in these mice, they have better exercise capacity, more mitochondria. And I believe there was a paper also where they looked at aging to show that the mice, had like better, less frailty and aging. There was one study that was nice. I think he did it in collaboration with Josh Sains, where they also looked at the NMJs, the neuromuscular junctions, to show that there are neurotrophic effects that help with reinnervation. And I think they did Duchenne's muscle dystrophy mouse models, where where PGC1 alpha was involved. So yeah. And irisin is, the leading secreted molecule where we have now a molecular handle when you now think about therapeutics, how to get to it, because you're probably aware that transcriptional coactivators are not very easy to, be targeted pharmacologically. And also, you try to be at the right level, right? Like the further downstream to get, the more things you influence, the broader the effects, but also, you might get unintended effects that you don't want, versus, and if you go down too specifically, maybe then you lose some of the effects that you actually want. So we hope that irisin is kind of in the comfortable Goldilocks middle there.
Yeah. And maybe this is a good time to mention brain-derived neurotrophic factor, BDNF. Now, previous work had provided evidence that, well, BDNF has an antidepressant effect. So exercise will increase BDNF levels. The most commonly prescribed antidepressants will increase BDNF levels. Electroconvulsive shock therapy will cause huge increase in BDNF.
Yeah.
Psychedelics, which there's a lot of interest now. So that's kind of a correlation, but there are data from animal studies that suggest that the increase in BDNF with these different interventions is necessary for the antidepressant effect. BDNF also plays important roles in learning and memory. So you found a connection between irisin and BDNF.
Yes, we did. So we did this in cultural experiments where we overexpressed FNDC5 or knocked down FNDC5 and showed that BDNF was regulated. And then I had mentioned the experiment where we peripherally delivered irisin or FNDC5 to increase concentration in the circulation. We also saw that BDNF levels were upregulated. I would like to say here, I don't think that BDNF is the only thing that irisin is working through. And I don't know if you're going to touch on later on our work in Alzheimer's disease, where we also see some, coming back to the immunology, some effects on immune cells, or on, astrocytes are not considered immune cells, but part of the cellular components of neuroinflammation. I think the one thing about BDNF, which, I always got asked, especially, is, they didn't really work out therapeutically. And I think my understanding is it's mainly due to a problem in delivery, like to get high enough levels of BDNF just in the brain or even in the brain region that you wanted it to have. And that's why I think, again, looking for other molecules that are either inducing BDNF or mimic BDNF, I think is also an interesting therapeutic options if those molecules have a better way to be delivered, or, in case of BDNF, I don't think the side effects were the problem, but some other, IGF-1 isn't one of these nice neurotrophic effects, but it has a pro-proliferative component, which is good if you think about neurogenesis. But in peripheral tissues, they're concerned about, pro-cancer, pro-growth effects there. So,
and that, BDNF, kind of the classical way of thinking of it is it's an activity-dependent neurotrophic factor. In other words, when neurons are firing, BDNF is released. And so, and we did some experiments that address this partly. We were looking at like the autonomic nervous system, and kind of bottom line of what we found is if we swamp the tissue with BDNF, it doesn't have beneficial effects. So as you mentioned, there has not only highly localized in a brain regional sense, but even in a neuron connection specific. What if, you got to see if irisin affects activity of neurons. What if irisin is kind of stimulating the neurons and that's increasing BDNF?
I guess depending, let's step back. So we have done now more work, in Alzheimer mouse models where we also delivered irisin. And it seems that, coming back to the exercise theme, that irisin gets to the regions of the brains it needs to go and has effects there. So we haven't seen, the same problem, which, as you reported, if you swamp the brain with BDNF, having problems there. It's a very interesting question how that works. And one thing that I'm thinking about is when we made these FNDC5 knockout mice, we didn't see a huge phenotype in these mice. If you have young group house mice, black 6 background, and you do a bunch of behavioral tests, there was nothing that really stood out. However, if you start challenging the mice, you make them exercise, you age them. We bred them with on an Alzheimer disease background, APP presenilin 1 mice. Then they showed cognitive deficits. Then they didn't have the learning that they needed. They had, faster cognitive decline than the wild type counterparts. So I think that irisin is a layer of stress protection that when you need to activate reserves, that's where it then comes in. And I think that makes sense also when if you look in the brain, in the young wild type mice that we exercise, the biggest effects we saw was on adult hippocampal neurogenesis, which, as you know, is an important mechanism of neuroplasticity. And it seems then when there was the challenge that you needed this additional neuroplasticity in exercise to adapt to this new situation, not having irisin was a problem. And in this case, we actually saw overgrowth of the neurons, that the trimming was lost that you need. If if students maybe remember, usually when you build a new neuron or new synapses, new circuits, there's usually more connections than you need. And then you trim it down to what is actually necessary. And if you don't do that, then there's a problem. You get too much activity in the wrong places. And that was some of the phenotype that we saw in these exercise mice. So that's where I think about irisin being important in these situations of additional stress.
Yeah, that makes sense. And that's kind of a common theme, it seems, when genes are manipulated, that oftentimes, you knock out a gene and the animal looks okay, but then when you subject them to some stressful condition or so, then you start to see something emerge. I'd briefly like to mention, so there's a few other factors that have been proposed to be either exokines is kind of the I don't know what you want to say.
moniker, the
yeah,
catchphrase, maybe you want to
yeah,
call it.
catchphrase, exerkines, molecules that are released from cells in response to exercise into the blood and have effects at a distance, I guess, almost like endocrine effects. So when Henriette von Prague, who, as you know, when she was in Rusty Gage's lab, she discovered that exercise increases hippocampal neurogenesis. She had this evidence that was surprising to me. There's a protein called cathepsin B,
Yeah.
which is, it's known because it's in lysosomes in cells, which are these acid bath compartments that are involved in autophagy and getting rid of things. But she found that in animals, and even I think she got blood samples from humans that exercised and found that in response to exercise, cathepsin B was increased in the blood. And then she had some evidence that it was mediating beneficial effects on the brain. So that's another candidate for an exerkine. You mentioned IGF-1. Something we were interested in a lot, particularly right before I retired, we did a lot of work with that's ketones. So, so fasting. Okay, so you're after you eat a meal, your glucose stores in the liver are replenished, and that glucose is always used before fats from your fat cells. And that takes, in the humans, 10 to 12 hours. And in mice or rats, it's actually much sooner. Within like even 4 to 6 hours, they start having elevation in release of fatty acids and elevation of ketones. And the ketones are an energy source for neurons, but they also have signaling functions. We found that one of these ketones, beta hydroxybutyrate, increases BDNF expression, and another lab up in New York found the same thing. So those are 4. Are there more than that besides irisin, cathepsin B, IGF-1, and beta hydroxybutyrate?
Yeah, there are a bunch of molecules. There's like some reviews just written about that. GLDP1 that came off Solvileda's lab, I think, is one of the ones. They showed it was released from the liver. What's his name? Tony Viscori at Stanford. I mean, he has published some work on in exercise. Was it VGF? I'm blanking on. Was it the VGF and VICAM1 molecules that I think it was VICAM1, which VCAM1 that they looked at. So there's several molecules out there, either protein based, that people are interested. I think when you think about translation, the problem is always you need to have efficacy in the brain, like it has to do what it wanted to do, but you can't have any side effects. And I mentioned, for example, IGF-1 is not good for that. I had talked with Henriette about cathepsin B. What's the status? But I don't, I don't recall. I think she might have mentioned that it was being looked at in the context of cancer as well. I don't want to say the wrong thing right now. I think other molecules are still in the race, but I know that, when I talked to Tony about I hope I now say the right molecule, that one of them also was not an option. So they were doing signaling on endothelial cells, and they had the same problem. It was stimulating endothelial cells and leading to growth, which had the side effect again of cancer. So that's a problem whenever you have something that stimulates growth, proliferation, that's good in the brain, and in the mouse where you only treat them for usually like whatever, a month, 2 months, 3 months. But if you think about you want to give it to a human, already an aged human, for maybe years at a time, then again, having anything that is related to cancer growth potential might not do that. And another area where people always look at is the heart. A lot of molecules end up having bad effects on the heart, and that's again obviously a a no-go. So there have been papers published on Irisin that actually show it's good in certain heart disease models. So, you know, fingers crossed. So so far we are still in the race. But that's the thing. It's not just, oh, you have to have this great molecule. It has to be safe. You have to be able to get it to the area of the brain where you want it to be in concentrations that are effective.
Yeah.
And you know, there's some, you know, I mean, pharma has been gotten much better to get molecules, you know, if you're across the blood-brain barrier, if you think about, you know, the antibody treatments now that they're looking at, and like looking, you know, harnessing different, you know, transporter proteins or carrier mechanisms across the blood-brain barrier. So yeah, but that's a different research area in itself. Like, how do we get big molecules into the brain?
Yeah, from the standpoint of general health and diet and lifestyle in the brain, though, the best thing you can do is exercise, not take any Irisin or IGF-1 or anything else.
Yeah.
And but you know, with aging and disabilities and so on, there are many people who can't.
Yeah.
You know, people who, for example, people who with paralysis.
Yeah.
You know, spinal cord injury or something like that, they can exercise. So if we can figure out, you know, can you kind of bypass having to do the exercise and still get at least some of the beneficial effects, that would be good.
Yeah. And you know, when I get asked about this point, you know, that's exactly what I usually also say. And the other thing is, you know, think about exercise. There's so many good things. I mean, we talked a little bit now about the cognitive effects. You have mentioned the mental health, stress reduction, anti-anxiety effects. And then we of course have all the effects on, you know, improving muscle strength, improving peripheral metabolism, improving cardiovascular health, you know, improving, you know, balance. You know, I mean, there's so many effects that exercise has. So the idea that there's one pill that gives you all these benefits, I don't know if that's realistic. But on the other hand, if you think about the population, you know, and not just paralysis, I mean, I just, you know, thinking about my own father who has a chronic disease, that, you know, people that have these conditions, they sometimes, if they can get to like 2,000, 3,000 steps a day, that's good for them. And to tell those humans to say, hey, why don't you just start running a mile or 3 miles a day? You should be fine in your dementia prevention. That almost seems a bit cynical. And the
Yeah.
same way how we treat, you know, metabolic diseases or other diseases, you know, you would prescribe medications if lifestyle modifications in itself were not effective. And then lastly, the point is we don't know how hard you have to push the system. And again, if you think about young, healthy people that can exercise a lot and where the brain is not that vulnerable yet, maybe that's fine. But it's also possible that if you talk about patients who already have developed, you know, mild cognitive impairment or even like early dementia, even maybe boosting irisin levels to what you can with exercise would not be enough, and you have to go in at pharmacological levels, right? That's you know, the other arm to think about.
Now can you talk give a little detail on the work you did you had a Nature Metabolism paper in 2021 on where you focus specifically on cognition and neurogenesis.
Yeah. So there are two things that we kind of showed. The one that I already have mentioned is the one is that, you know, lack of irisin led to, like, you know, impaired cognitive function in mice, that you, like, if you don't have irisin and you exercise, you don't get the improvements in spatial learning and benefit you usually get in exercise. If you age the mice, their cognitive decline is faster. If you have them on an Alzheimer background, their cognitive decline is faster. And then we traced that back, specifically the exercise modulation, about modulating adult hippocampal neurogenesis, and we kind of touched on that. The other part what we did is we looked at the therapeutic portion of it, and now I have to move just one over because the sun's coming around. Let's see if we can get out of the sun. You know, it's too sad, like on a January day. Is, you know, can you use irisin therapeutically? In this case, we choose Alzheimer's disease because we know iris or exercise effective in Alzheimer's disease, and we still don't have that many effective treatment options, and none based on exercise. And there we showed that giving irisin peripherally, and this time, which was also a pretty big deal in the paper, is we were able to just give the irisin part. So thinking about biotech and drug development, if you can figure out which is the active part of the molecule and you base your drug on that, you're much closer to getting to clinic than if you have to work with a large, type 1 membrane protein. How do you express it? How do you purify it? So we showed just giving the irisin part in to these Alzheimer mice, slowed down the cognitive decline, and in part by modulating neuroinflammation. So we looked at microglia and astrocytes. The receptor, the putative receptor for irisin, is highly expressed on astrocytes and microglia, and we showed actually with a storm super resolution microscopy assay, actually we could show that irisin is binding on these astrocytes. So that was the other kind of big part. And there has been now follow-up work in Rudy Tanzi's lab and Sehun Choi's lab, which I was a collaborator on, where they use now these 3D Alzheimer human culture models, Alzheimer in a dish, to also show that part of the effects were through astrocyte regulation, kind of, confirming the data that we had. And there's another work from Ted Dawson's lab, and Bruce Spiegelman. I'm also a collaborator on that in the Parkinson's disease mouse model, where we also showed it was neuroprotective there, which is
and there,
good to see.
that one, okay, so with Rudy Tanzi and the organoids, brain organoids, the irisin increased the production of neprilysin, an enzyme that breaks down the amyloid protein. Is that?
Yeah.
And that was in astrocytes. And then with Ted Dawson, who's a colleague of mine at Hopkins, in that case, so that was a PNAS paper, right? And and they're kind of, as far as mechanism goes, there was evidence that irisin might be stimulating autophagy.
Yes, and there's a follow-up, or not a follow-up paper, how do you say, another paper in Drosophila model, where they also showed that irisin could be a regulator of autophagy. So, which is, kind of for the people that work in model organisms, I think this is a very interesting discovery. And they actually made that exercise in the Drosophila flies as well. I forgot how they did it. I think maybe they climbed a ladder or something. I forgot.
Yeah, they can evaluate motor function and cognition in the fly model. How big is irisin? How many amino acids?
It's 112 amino acids. If it's not glycosylated, it runs at 12 kilodaltons roughly. But there is glycosylation sites, and it gets glycosylated. All the cells we express irisin, we saw glycosylation, so then it usually runs around 24, 25 kilodaltons.
And has anyone tried to take different fragments of that 100 and some amino and see if a smaller one is active?
I don't think, not that I'm aware of. I think, because from the pharma point of view, I think the irisin that we have is pretty small already, and it seems to go and do the things that we like. But I'm not saying that maybe that in future protein engineering, maybe that's something to look into.
But
if
you
you
have
can
to
even break it down smaller.
You produce it in what system do you use to make recombinant irisin?
Usually people use either 293 cells or CHO cells.
Because
That's usually when you talk
big
when you talk about like pharma grade or like large quantities. That's usually the cultural system where you can push it. If you think about doing it in primary cell culture systems, usually you can. I mean, they want to grow them by the bucket, right? So
yeah. And it's a little too big to synthesize, I think.
Yes, I think I mean, I think you might be able to do that, but then you would not be able to have the glycosylation sites. I'm not a peptide biochemist. So a colleague of mine has mentioned it might be possible, but then the question is, if you don't have the glycosylation,
right,
is it as active, as having the glycosylation? So that's the caveat. And I think it would be much more expensive to do that than versus doing large scale productions in these
Yeah, that's
cell types.
okay. And then what the receptor for irisin, it has not been identified. You had some evidence for an integrin maybe being.
Yeah, there was a paper in Cell, in 2000 and was it 16, 18, I think, which was like the tail end of my postdoctoral work and then has been taken over by a new talented postdoc in Bruce's lab, where they showed that integrin receptor, and specifically alpha V, probably beta 5, was a putative receptor for irisin in bone and fat cells. And then we showed that irisin in neurons is also binding alpha V beta 5 integrins, but not alpha V beta 3. We used that as a control to say, we want something that is very similar, but it's not the active receptor. And there's another work from Dr. Spiegelman's lab in molecular cell from, I think, was it this year or last year, where they also tried to go more on do a lot of receptor characterizations, and they postulate that there might be even a cofactor binding to the receptor in these systems.
And then, so irisin binds to a receptor, maybe in this integrin, and then what happens inside the cell that, for example, leads to increased BDNF, turns on the BDNF gene?
So yeah, Sehun, in the neuron paper, they also, in their 3D cultures, they were able to, stimulate irisin. They use different neutralizing antibodies or receptor inhibitors. And the classic signaling cascade that gets turned on downstream of integrin signalings is a phosphorylation of FAC, PFAC, and then CREB, which, if you think about BDNF, is one of the upstream factors there as well. And then they further down possibly identified, neprilysin was one of the, targets regulated, and other cytokines that then might help also maybe in the crosstalk with microglia. So the receptor, if you look at alpha V beta 5, is very high expressed on astrocytes and microglia, which kind of would fit into that profile. There's also a possibility that, in stress situations, maybe in neurons, it gets more upregulated, and then you would see more binding there. So I think there's still, if you ask me, more work to do to kind of understand, especially in the in vivo context. So there's a lot of really nice work in culture or in in vitro systems, which is great because you can really, dissect on a molecule by molecule level, like what's going on. But I think we also need to understand it better in vivo, and that's work that is happening in my lab to understand in an intact mouse, where's irisin binding at which cell types, how does it change in different diseases, and then as you said, what where does it go? I think there's more work to be done to kind of completely understand, and we have talked about different mechanisms, BDNF, neuroinflammation, autophagy. So I think they might also be related. I mean, there's also work that shows that inflammation and autophagy might be connected. So, I mean, this is the part where, you can see I get excited about it, but I think this is where I see, or where in my lab, there's a lot of work on understanding more on the mechanistic level. We're also looking in other neurodegenerative diseases to see what it's doing there. Some more translational work, to understand how we can use irisin as a drug. So, I mean, there's still a lot left to do, but it's quite exciting work. And so far, what for me is reassuring is that there's a bunch of labs that finding pieces that fit together. So that I find that very encouraging. And I always get asked by journalists, "Oh, when do we have the drug?" I don't know. It takes a long time, but I think so far we're on track. Hopefully the journey has a happy ending.
Yeah, it's very exciting times. I was thinking, so integrins, I mostly know about integrins because they bind to certain extracellular matrix, or some of them anyway, like laminin, for example. And then you mentioned that there's, excuse me, some evidence that there may be some cofactor involved. So I know that for fibroblast growth factor 2, FGF2, and I think some other growth factors, they have to interact with heparan sulfate proteoglycans on the surface. And so the idea there is like, if a growth factor is released from a cell, it like FGF2, it's not just like diffusing, it's binding to something very soon and staying there.
Yeah.
Um,
I know this from the efferent receptor
uh,
interaction, which, again, if you think about, axon guidance, I think it's a similar principle that, that synapse or that gets activated. Now we want a stronger connection here and not, everywhere. Like this is like, how do you get targeted, improved connectivity? Yeah.
yeah.
Very fascinating how that works.
And have you or anyone looked at if irisin, so you mentioned the PGC1 alpha connection, but will irisin itself, oh yeah, you mentioned this, will irisin increase the number of mitochondria in muscle cells? Is that true?
They found it in fat cells. So it's made in the muscle, it travels in the blood, and then
Uh,
it activates a fat. So we have different kinds of fat, white fat, brown fat, depending on the mitochondrial content. And some of the, especially the subcutaneous fat, can get activated to become more brown. That's what they call it, a beige fat. And that's why it's called beige, because as you know, the mitochondrial content is what gives it the browner color. And that was the original Nature paper where they showed that there was an increase in mitochondrial signature in that. And they showed that in exercise there's more browning of the white fat, and then they showed that irisin was at least one of the sufficient to that.
That involves then increase in the amount of the uncoupling protein in the mitochondria?
Yes,
right?
that's what they showed. UCP1 was upregulated in inguinal fat, or like the culture cells that they looked at.
That would be interesting to look at in neurons. We did some work on uncoupling proteins in neurons, and also there's a company that makes this is an old drug, a mitochondrial uncoupler 2,4-dinitrophenol, DNP. And it's actually very neuroprotective at doses that don't cause any major side effects. So I guess the point there is, and we think it's actually inducing a stress response in cells, because when you uncouple oxidative phosphorylation, the cells initiate like a stress response, but it's good, kind of like exercise induces. So I think it would be interesting to see if irisin affects uncoupling proteins in neurons and what else? Oh, and mitochondrial biogenesis.
Yeah, I think in the gene signature that we looked at, I mean, that's something we are kind of trying to tease apart. And if you look at these gene signatures, if you look at Alzheimer's disease and Parkinson's disease, a lot of the gene sets that are curated, mitochondrial genes or oxidative phosphorylation is in there as one of the driving factors. And I think, yeah, as you just described, energy demands can be met, waste products can get out. So it kind of hangs together. Yeah.
And another one, we found that exercise will increase levels of a protein called sirtuin-3, which is a mitochondrial protein deacetylase that seems to be very important for mitochondrial health and function. And it would be interesting to see if irisin can explain that. And that was, we looked in vivo and we had SIRT3 knockout mice and so on.
I mean,
yeah, but that was very interesting because we found that it was like a cell metabolism paper, I can't remember, 2016 or something. And so kind of the bottom line was that if we blocked NMDA receptors, glutamate receptors, then exercise no longer had caused an increase in SIRT3 and had neuroprotective effects. So that was kind of interesting too.
Do you think it's because the if the neurons can't meet the increased metabolic demands that's a problem, or that they couldn't, or do you think they needed that to exert the
no, I think it's just
neuroplasticity, or?
kind of evidence that exercise is increasing neural network activity
Yeah.
in the brain, and if you suppress that pharmacologically, then some of the benefits of exercise are lost.
Yeah.
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 So.
Yeah.
Yeah.
So there's one interesting study I we mentioned.
Sorry, I keep one simple experiment. Just
Yeah.
give Irisin to animals, kill them at different time points, and do staining for CFOS. Just
Yes.
look, is there any, that's kind of a crude, but
Yeah. So what we did show is that in the knockout mice, so we had the Irisin wild type and the knockout mice, we did make them exercise because that activates newborn neurons, and it activates specifically also CFOS, and it activates neuronal activity, which you can read out by CFOS. So, and we did actually show that in the knockout mice there was aberrant CFOS expression, and thereby we induced or deduced that also neuronal activity, as I said, because at that time, it was also during the COVID lockdown. It was just not possible for us to do, EFIS on slices, or to do like calcium imaging. So as you said, the one thing that you can use is markers for neuronal activation. We choose CFOS, and we did the exercise model because we want to specifically get to the newborn neurons. To
neat.
see what's happening there. So yeah, that was a really nice experiment. One interesting experiment that I always kind of like to bring up when we talk about what are the systemic effects versus just neuronal activation is Henriette van Praag's lab and Justin. God, no, I'm blinking on the name. He's also in Urbana-Champaign. What's his last name? He's was in the same institute where Art Kramer was. It'll come to me in a minute. So they also did exercise studies, and what they show is basically the running part is important for this neurotrophic and cognitive function effect. So the experiment that Henriette did is not just the how do you say the neuronal activation. So what they did is they had 4 different conditions. They had standard housing, no wheel. They had the housing with the wheel, and then they had the enriched environments, which, as you know, one of the things they do is increase neuronal activation. So an enriched cage is usually a cage filled with little toys, things for the mice to climb on, things to interact with, maybe like other things in there, so there's more enriched environment. But it usually includes also a running wheel. And so they had then the condition enriched environment with running wheel and enriched environment without a running wheel. And then they show that as soon as you remove the running wheel, the effects on increasing neurogenesis were blunted. There was still a small increase in neurogenesis. It was not that there was nothing there, but much less. And just why can I not remember his last name? I'm sorry. I'm bad with names. If you listen to this,
I am too.
I remember all your research, but I can't remember your last name. They looked then at cognitive function in this case, I think in the CFC assay, that shows that yes, there might be also a neuronal activation component, but there's also something about exercise, the running, what does change things. And one of the things that we were thinking about coming from a muscle-centric perspective is, of course, muscle activation secretes all these, myokines, and there is systemic effects, and also in systemic energy demands and what gets secreted, or what are the metabolic demands on the body, which is very different, right? If you run in a running wheel versus a mouse, exploring an enriched environment. So this was again one of the reasons why I was going after myokines in the place, because I was like, okay, this shows that there's something to it. And I guess also, and one of the reasons why Henriette, we are very good friends, looking at this phenomenon.
Yeah. Okay, great. I guess we can wrap up now. And so what we learned is that there's a lot of work going on trying to understand how exercise benefits the brain in terms of improving mood, cognition, maybe protecting the brain against aging and age-related disorders like Alzheimer's, Parkinson's, and so on. And mechanisms are being elucidated in animal models, and translation to humans is always a little harder, but there is actually work going on there. You mentioned Tony Westcore. You know, and I had him on a podcast too a while back. And so he did all these blood transfusions, showed that if you take blood from young animals and you put it in old animals, it improves their cognition. If you take blood from animals that are exercised and put it in, I guess, old animals, it's beneficial. And I'm not sure what the status is. They're doing clinical trials in humans. And
in Norway, they have an ongoing clinical trial at the University of Trondheim where they're doing blood transfusions, I think in Alzheimer or mild cognitive impaired from young or even young exercised, taking blood or plasma. It's usually blood, it's plasma from young there. So Tony and I, we always have this interesting conversations, do you need to find the one factor, or should you go with the cocktail that you have in the plasma, right? And I think there benefits to both of them. I think one of the problems is just if you want to maybe you can get away with plasma, but if you want to pharmacologically boost one component, you would like to know which one it is, and you're more specific. But I think that's very interesting work that he has done, and he has done a lot to also make the field aware of, yes, there are transferable effects in the blood, because you can imagine whenever you start out in a new field and talking about secreted factors in the blood that go to the brain, we were met with a bit of skepticism. So I think his work, and I mentioned Saul Villada's lab, I think very, very important work to kind of show, yes, there are these mediators in the blood, and if you can find them, there's at least a good chance that we might turn them into a drug.
Yeah, I don't know if you have children, but we have grandchild now, and so I'm thinking that I should bank some blood from our kids or our kids after they've exercised for a while, and then in case I need it when I get a little older.
I have a daughter. I have not had that thought, but so I'm still able to do my exercise. So I'll maybe later. But I think these are interesting studies, and see how that how they pan out. I think it's still ongoing. I don't think they have released results yet from the Norway study. And
I haven't seen anything published yet.
Yeah.
Okay, Christiane, I enjoyed meeting you and talking with you. I followed your work, very exciting, and I'll try to keep track of how it progresses in the following years.
Thank you. I had great fun in this in our discussions. I think it's very stimulating, and kind of like the sciencey part. I mean,
it's almost like a two-person lab meeting, right?
Yes, it is. And I think we can always see that, when you talk to colleagues that especially when they're a little, not completely, when a little bit on the other side, like the kind of how do you like cross what do they call it, cross pollination that is happening, and how, I mean, if we discussed so many new research ideas, I just need to get another 4-hour ones to do all of them. But I think we have I think we identified a lot of really important questions that need to be answered, and I think we have the tools to do that. So it was a great pleasure to talk to you today, and I hope that our audience also will enjoy our little chat that we had as much as we enjoyed it.
I expect they will, and they should go out and exercise before they listen to it. They'll maybe enjoy it a little more and remember more of it.
Yes.
Okay, Christiane. Bye.
Bye. Take care.